All right. Hey, everyone. Welcome back. I am so excited for this deep dive. We're going to be talking about one of my favorite topics, antibiotic resistance.
Yeah.
Specifically, we're going to really get into the mechanisms that bacteria use to basically outsmart these really powerful drugs.
Yeah.
And we're going to be taking a lot of our insights from Antibiotics, Challenges, Mechanisms, Opportunities by Christopher Walsh and Timothy Wenswich.
Great book.
Yeah, it's a really good one. So, as a medical microbiologist yourself, I'm sure you've encountered this in your work. It really is kind of like this constant arms race, right?
It is, yeah. It's constantly evolving. It's incredible how these organisms, they've been around for billions of years and they've figured out a way to survive almost everything we throw at them.
Exactly, yeah. And I think one of the things that I find just so fascinating is this idea of target modification.
Yeah.
It seems like such a proactive thing. Like
Yeah.
they're not even waiting for us to attack them. They're just like, oh, I know what you're going to do. I'm going to change this.
Right. It's like they've been studying our playbook. I mean, instead of waiting for the antibiotic to do its thing, they preemptively change the structures that the antibiotic needs to bind to. It's brilliant.
Yeah, it's like they're changing the locks before we can even get there.
Precisely.
Yeah.
So they're tweaking their cellular machinery just enough so that it throws off the antibiotic. But, and this is the key, they're still able to function normally.
Right. It can't affect them negatively or they wouldn't do it.
Exactly. And one classic example, we see this all the time, is Staphylococcus aureus or staph. They can modify DNA gyrase. So this is an enzyme that's essential for DNA replication, and it's the target of fluoroquinolones. But staph can just tweak it slightly, just enough so that the fluoroquinolone can't bind effectively.
Right. That's why we always have so much trouble with those staph infections. They just keep coming back.
Exactly.
So Walsh and Wenswich talk a lot about mutations. And, I always think about mutations as these like kind of big, scary things.
Mm-hmm.
But a lot of times when it comes to antibiotic resistance, it can be really small, subtle changes.
Yeah, absolutely. I mean, mutations can be as simple as a single change in a DNA base pair.
Yeah.
And that can lead to, a single amino acid change in a protein. And even that tiny change, that can be enough to prevent the antibiotic from binding.
So it's this like really delicate balancing act for the bacteria, right? They have to change enough to resist the antibiotic, but not so much that it affects their own survival.
Yeah, it's like walking a tightrope, and they do it remarkably well.
They do. What are some of the examples that you find just really kind of stand out?
Well, one that always comes to mind is Mycobacterium tuberculosis, so TB.
Yeah.
Rifampicin is a really important drug for treating TB, but TB has become increasingly resistant to rifampicin, and that's largely due to mutations in a gene called ARPOB. And this gene encodes a subunit of RNA polymerase, which is the enzyme that makes RNA. So the mutations change RNA polymerase just enough that it's still functional for the bacteria, but it's no longer sensitive to rifampicin.
Wow. So they can still function. They can still transcribe their genes.
Right.
But they've just changed enough that that drug can't bind anymore.
Exactly.
It's incredible.
Yeah.
Another tactic that I thought was really interesting that Walsh and Wenswich talk about is methylation. And to me, that always seems like a much more kind of subtle way of doing things.
It is, yeah. It's like they're trying to fly under the radar. So with methylation, the bacteria basically add a methyl group. And they do this to specific sites on their ribosomal RNA. And by adding that little methyl group, it's enough to prevent certain antibiotics from binding to the ribosome and blocking protein synthesis.
So it was like they're disguising themselves.
Exactly.
Yeah, that's a good way to put it. Like, putting on a little hat and glasses and hoping nobody recognizes you.
And it's a very effective strategy. So, for example, methylation of 16S ribosomal RNA can confer resistance to a whole class of antibiotics called aminoglycosides. And these include drugs like kanamycin and gentamicin.
Right. So this one little change can affect a whole class of drugs. It's incredible.
Exactly.
And what I found really interesting is that, this isn't just a tactic that's used by pathogens.
Right.
Like even bacteria that produce antibiotics, they use methylation as a way to protect themselves from their own weapons.
Yeah, I mean, it makes sense if you think about it. If you're producing something that's toxic, you need to make sure you don't poison yourself.
Yeah, it really speaks to like how ancient these mechanisms are.
Yeah, absolutely. I mean, this has been going on long before humans even walked the earth.
Right. It's not just like something they developed because we came along.
Exactly.
All right. So let's switch gears a little bit. Let's talk about something that, to me, it feels a little bit more aggressive.
Okay.
And that is this idea of electrostatic warfare.
Yeah. So this is a really fascinating strategy, and it all comes down to charges. Opposite charges attract, like charges repel. So bacteria have figured out a way to manipulate the charges on their cell surfaces to basically repel antibiotics.
So they can literally like change their surface charge.
Yeah. And this is especially effective against antibiotics that have a positive charge. So these are called cationic antibiotics. And these antibiotics are attracted to the negative charges that are normally found on bacterial cell surfaces. So the bacteria, they're like, okay, I'm going to get rid of those negative charges. I'm going to mask them. I'm going to add some positive charges. And they do this by adding, different chemical groups to their cell envelope components.
It's literally like building a shield.
It is, yeah. And a great example of this is how gram-negative bacteria modify a molecule called lipid A. So lipid A is a component of lipopolysaccharide, which is a major part of the outer membrane of gram-negative bacteria. And they modify lipid A with a molecule called 4-amino-L-arabones. Now, that's a mouthful.
It is.
But the important thing is that this modification adds positive charges to lipid A, which helps to repel polymyxins. So polymyxins are a class of cationic antibiotics. So by adding those positive charges, the bacteria are basically saying, no, you can't come in here. You're not welcome.
Right. Like, get away from me.
Exactly.
It's incredible. So, one of the things that I thought was really interesting that Walsh and Vensfis go into detail on is vancomycin resistance.
Oh, yeah.
Particularly in enterococci.
Right.
Or VRE. And, vancomycin, I mean, this is like our big gun, right?
It is. It's one of the last resorts for treating serious infections.
And enterococci have figured out a way to resist this.
They have, unfortunately. And it's a really clever mechanism. So normally vancomycin binds to the diallo-diala terminus of peptidoglycan precursors. And peptidoglycan is what makes up the bacterial cell wall. So it's essential for the bacteria to survive. But enterococci that are resistant to vancomycin, they have this system called the Van Hack system. Yeah. And this system allows them to modify the diallo-dial terminus.
So they're changing the target itself that vancomycin normally binds to.
Yeah, exactly. They're like, okay, you want to bind to this? Well, guess what? It's not there anymore. And so what they do is they swap out one of the D-alanine molecules for a D-lactate molecule. And by making this one little change, it dramatically reduces vancomycin's binding affinity.
Wow.
Yeah. And so vancomycin can't bind to the peptidoglycan precursors. It can't inhibit cell wall synthesis. And the bacteria survives.
It's amazing how just that one little change can make such a difference.
It is, yeah. It's a testament to, how evolution works, Just a small tweak can make all the difference in the world.
It's a reminder that evolution doesn't have to be this like big dramatic thing.
Right.
It can be these really subtle, tiny changes that have this huge effect.
Absolutely.
One of the things I thought was really interesting about the Van Hack system is that it probably originated in the bacteria that produce vancomycin.
Right. This makes perfect sense. If you think about it, these bacteria, they're producing vancomycin to kill other bacteria. So they need to protect themselves from their own weapon. And so they have this system, the Van Hack system, that allows them to do just that.
So they develop the shield before they develop the weapon.
Exactly.
That's fascinating.
It is, yeah. And it highlights, the long history of this arms race. Bacteria have been fighting each other for billions of years.
Right. This isn't a new thing. It's something that we're, kind of just now understanding the complexities of it. Speaking of, resistance, let's talk about MRSA.
Okay.
Methicillin-resistant Staphylococcus aureus. This is like the poster child for antibiotic resistance.
It is, yeah. It's, it's a major problem in hospitals.
Yeah, and it seems like every year, every day, they're getting harder and harder to treat.
They are, yeah. And, one of the main ways that MRSA resists methicillin and other beta-lactam antibiotics is by, essentially swapping out a sensitive target for a resistant one.
Like target swapping.
It is, yeah. It's kind of like, if you have a weak spot, just replace it with something stronger.
Right.
And so what they do is they acquire a gene called MECA. And this gene encodes a modified penicillin-binding protein, and we call it PBP2A. And this altered protein has a much, much lower affinity for beta-lactam antibiotics.
So instead of just modifying their existing penicillin-binding protein, they're bringing in this whole new protein.
Exactly. Yeah, they're just bringing in a specialist. And this specialist is really good at resisting beta-lactam antibiotics.
That is so incredible to me.
It is.
So, and then to make matters worse, this MECA gene it's often carried on mobile genetic elements.
Right. Yeah, these are called SCC-MEC cassettes. And they can be transferred between different staph strains, and that's why methicillin resistance has spread so rapidly among staph.
Right. So it's not just like one strain that has to develop this resistance.
Right.
It can literally spread to other ones.
Exactly.
But we are making some progress, right? I mean,
Yeah.
there are these fifth-generation cephalosporins that are starting to be used more.
Right. Yeah, and these are really promising. So these are cephalosporins like ceftaroline, and they have a different binding strategy than earlier cephalosporins. So earlier cephalosporins, they only bound to the active site of PDP2A. But these fifth-generation cephalosporins, they can bind to both the active site and an allosteric site.
So like a double whammy.
It is, yeah.
They're finding a new way to actually target this protein.
Exactly. Yeah, they're figuring out a way to overcome those structural changes that make PBP2A resistant to other beta-lactams.
So it's like finding a new key for a lock that's been changed.
Exactly.
There's also some, other really exciting stuff happening, right? This research into non-beta-lactam PBP2A inhibitors.
Right. Yeah, this is cutting-edge stuff. So researchers are using computer simulations to screen vast libraries of compounds, and they're looking for molecules that can inhibit PBP2A. But, and this is the key, they're not beta-lactams. So these molecules have completely different chemical structures.
It's like finding a back door.
It is.
Like they're trying to find a whole new way to target this protein.
Exactly. And it's really exciting because it opens up a whole new avenue for drug development. I mean, instead of relying on beta-lactams, which MRSA has become so good at resisting, we can now target PBP2A with something completely different.
Yeah. It's almost like a new era in the fight against MRSA.
It is, yeah. And it really highlights, the power of computational approaches. These approaches allow us to screen millions of compounds, in a very short amount of time, and we can identify promising leads that can then be tested and optimized.
That's amazing.
It is.
I love that we're able to use all of this technology to kind of fight back against these really, amazing bacteria.
Yeah, I mean, you have to admire their ingenuity. But we're not giving up.
We're not. So I think that's a really good place to stop for this first part of our deep dive. We've covered a lot of ground, but we've only just scratched the surface.
Exactly.
In the next part, we're going to delve even deeper into some of these resistance mechanisms. We're going to talk about some of the really specific examples. And I think we should end with a little quiz.
Oh, fun.
To see how much you've learned. So stay tuned.
Yeah, this is great stuff. I'm really enjoying this conversation.
Now, too, I love talking about that.
Yeah.
All right, so we'll see you in part 2.
All right. Okay, are you ready for the first question?
Yeah, hit me.
All right. Which bacterial enzyme is responsible for adding lysine to phosphatidylglycerol, contributing to daptomycin resistance in MRSA?
Okay. Lysine, phosphatidylglycerol, daptomycin resistance. I'm going to go back to what we were talking about with electrostatic warfare.
Okay.
And how MRSA changes its surface charge.
Mm-hmm.
I feel like that MPRF enzyme has something to do with this.
You are absolutely right. That is the one. MPRF is the enzyme that links lysine to phosphatidylglycerol.
Right.
And then it flips this modified lipid to the outer leaflet of the membrane. So it's basically decreasing the negative charge on the surface, making it harder for daptomycin to bind.
So it's like building a shield against daptomycin specifically.
Yeah, exactly. It's like a custom-made shield.
That is so cool. All right, what's question number 2?
Okay, let's see. What chemical modification does the CFR gene product catalyze, leading to linezolid resistance in some bacteria?
Okay, CFR gene, linezolid resistance. I'm going to go back to our discussion of methylation.
Okay, good.
I remember that methylation can target different sites in ribosomal RNA.
Mm-hmm.
And I think the DEPR gene, if I'm remembering correctly, encodes a methylase that modifies the 23SR RNA.
Yes.
Which then leads to linezolid resistance.
That is correct. You got it. The CFR gene product, the CFR enzyme, it catalyzes the methylation of a specific adenine. It's 82503 and 23SR RNA.
Okay. So again, modifying the ribosome to block the antibiotic?
Yeah, it's all about making those little tweaks to prevent binding.
It's amazing how targeted they can be.
Right.
All right, what's question number 3?
All right, this one's a little bit more complex. Explain the mechanism by which the Van Hack system confers vancomycin resistance in Enterococci.
Ah, the Van Hack system. Okay, so this is all about changing the target that vancomycin binds to, right?
Yes, precisely.
So if I remember correctly, they swap out D-alanine for D-lactate in the peptidoglycan precursor.
Yes.
So Van H makes the D-lactate. Van A then hooks it up to D-alanine.
Yeah.
And then Van X cleans up any D-ala that's still hanging around.
Perfect. You nailed it. So they end up with this D-alidine lactate terminus instead of the D-alidiola, and vancomycin just can't bind to it.
So the bacteria has essentially changed the locks.
Exactly. Vancomycin can't get in. It can't do its job.
It's so clever. Okay, 2 more questions. What's next?
Okay. Why are 5th-generation cephalosporins more effective against MRSA than earlier generations?
Okay, back to MRSA and that tricky PP22A. So we talked about how these newer cephalosporins bind differently, right?
Yeah.
So earlier generations, they only targeted the active site of BBP2A. But the 5th generation cephalosporins, like ceftaroline, can bind to both the active site and an allosteric site. So they have this dual binding action, which means they can still inhibit PVP2A even with those structural changes that usually cause resistance.
That's a great explanation. You're exactly right. That allosteric binding site is key.
Yeah, it's like finding a new way in. Okay, last question. Bring it on.
All right. What makes the discovery of non-beta-lactam PVP2A inhibitors significant in the fight against MRSA?
Ooh, this is the exciting one. So instead of trying to use beta-lactams, which MRSA is so good at resisting,
Right.
we're basically finding completely new molecules that can inhibit PVP2A.
Yeah.
Like a whole new approach.
Yeah.
And MRSA hasn't had time to develop resistance yet.
Exactly. We're essentially bypassing those well-established resistance mechanisms by targeting PVP2A with a completely different type of molecule. And the fact that these new inhibitors were discovered using computational approaches, that's huge.
Yeah.
It's like we've opened up a whole new world of possibilities for drug development.
Yeah.
I mean, we can screen millions of compounds in silico, identify the most promising ones, and then optimize them.
It's amazing how technology is helping us in this fight.
It really is. It gives me a lot of hope for the future.
Me too.
Yeah.
It's like we're finally starting to catch up to these bacterial masterminds. I mean, it really is amazing when you think about it, like how much these bacteria have evolved to resist what we're throwing at them.
Oh, yeah. It's incredible. And it just shows, how dynamic this field is. We're constantly having to learn and adapt because the bacteria, they're not standing still.
No, they're not. They're always one step ahead, it feels like.
Yeah.
Like we think we've got them figured out,
Yeah.
and then boom, new resistance mechanism.
Right. It's a constant back and forth. And that's why it's so important to keep researching and developing new strategies.
Absolutely. And I mean, we've talked about just a few of the tactics that bacteria use. Modifying their targets, that electrostatic warfare, changing their surface charge, even just swapping out vulnerable components for ones that are resistant to our drugs.
Yeah. It's amazing how adaptable and resilient they are.
It really is.
Yeah.
I mean, you got to kind of admire them a little bit.
Yeah.
But at the same time, we have to fight back.
We do. And it's not just about developing new antibiotics, although that's obviously a crucial part of it. We also need to be smarter about how we use the antibiotics we already have.
Right. Prevent the spread of resistance in the first place.
Exactly. And improve our diagnostics so we can target treatment more effectively.
Right. So that we're not just throwing antibiotics at everything and hoping something sticks.
Exactly.
Well, I think this has been an incredible deep dive. And, I hope that for all of our medical microbiologists out there listening, you've maybe learned something new. Or maybe it sparked some ideas for your own research, for your own clinical practice.
I hope so, too. It's a fascinating field, and there's still so much we don't know.
Exactly. And that's what makes it so exciting. And, if you want to really, really dive deep into this, Antibiotics, Challenges, Mechanisms, Opportunities by Walsh and Vencevich is a fantastic resource.
Oh, yeah, that's a great book. Really comprehensive.
It really is. So until our next deep dive, keep exploring, keep asking questions.
Absolutely. Never stop learning.
The bacteria certainly aren't stopping.
No, they're not. And they'll continue to surprise us, I'm sure.
I have no doubt. Well, thank you so much for joining me on this journey.
It's been a pleasure.
And to all of our listeners out there, thanks for tuning in.